So our next portion that we're going to talk about is actually a little bit about affecting that action potential and affecting how those signals are transmitted across the length of a neuron. So we're going to be talking about what are called the nodes of Ranvier, and these are related to the myelin sheath, which we've actually talked about very, very briefly previously. So we mentioned the myelin sheath as being made up of glial cells, our support system for our neurons, and they're forming these little hot dog bun looking things that run along the length of an axon. And now the nodes of Ranvier are actually the gaps that exist in between our glial cells, in between the little pieces of our myelin sheath. And so our nodes are going to be blank spaces where the surface of the cell, where the surface of that axon is still exposed to the area outside of the cell. And we can draw a blown up picture of it here. So we can have our glial cells, and they come very close to each other, but they don't quite touch. And they're going to continue onward, so on and so forth. And so these little spaces in between here, these are our nodes of Ranvier. And so our myelin sheath and these gaps between the parts of the myelin sheath are actually very functional. So we had talked about the myelin sheath as being there as sort of a insulation, where it's protecting and wrapping around our axon, and the gaps are then where the axon is left to be fully functional. And these gaps allow our conduction of action potentials to actually skip ahead. So if we think back to where we were talking about an action potential happening, and in the surface of our axon, in that cell membrane, we have those channels that allow sodium and potassium ions in and out. When we have these glial cells, when we have this myelin sheath right up against the axon, it's actually covering the surface of the cell. And it means that there aren't any channels here. So we can have an action potential when the surface of the cell is open. So we can see our really poor action potential there when it's open. But where it's covered, where these myelin sheath cells exist, you can't have an action potential triggered. And so what happens is all of the ions that have flown into and out of the cell, so we have our abundance of sodium ions inside from firing over here, those sodium ions can move ahead, but they can't trigger an action potential in this space. So they kind of have to move forward. They skip the section where they can't trigger an action potential, and they trigger one at the next available space, which is one of these nodes of Ranvier. So as soon as we hit this node here, we can trigger a new action potential. And then this action potential leads us to have more sodium ions inside again, and they can't trigger when it's covered by the myelin sheath, so they have to move ahead. And so we can only get our action potentials in these gaps. And this is actually a time-saving mechanism, so we can conduct that signal much faster by skipping over all of the areas that have myelin sheath. So instead of having to fire, see if we can get rid of some of this mess here, instead of having to fire at every single part of this of the cell here, so instead of firing here and here and here and here, because of this insulation, because of the existence of that myelin sheath, we only see firing where there's no myelin sheath. So we skip all of this, and then we can go here, and we can skip here and fire here. And so with less individual action potentials having to happen across the whole length of this axon, by skipping all these portions, the signal actually travels much faster. So we skip ahead, and we get these action potentials traveling infinitely faster. Well, not infinitely. I suppose we can calculate how much faster. But this is how we can have a signal traveling across very, very long neurons. And in fact, we have neurons within our body that stretch for very long distances. So the longest one that we have travels from our leg all the way into our spine. So it is more than a meter long in a lot of people. And if we had to have these action potentials, this firing occur every single step of the way, then it would take a very long time for you to tell if you have, say, stubbed your toe. But by having this extra insulation, by having these signals skip ahead, we can still have a relatively quick response time, even in a very, very long neuron, because that insulation and those gaps between the insulation lets that signal move much quicker. So now that we've talked about the neurons themselves, we've talked about a singular neuron, but of course that's not the whole picture. We're going to have multiple neurons, hundreds upon hundreds of thousands of millions of neurons. And so they have to connect somehow. And so originally, when people first started looking at cells, and when they started figuring out that neurons were somehow involved in how our biological processes were happening, people thought that one neuron connected directly to another. They assumed that they made physical contact. However, our neurons don't actually make physical contact. There's a small, teeny tiny gap in between neighboring neurons, and this gap is called the synaptic cleft, or sometimes you'll see it called a synapse. And that is the space from between the axon, those axon terminals, the end of the axon in one cell, and the dendrites, the beginning of the next cell. And so these synapses are going to be a functional but not physical connection between neurons and their target. Usually a neuron and the next neuron, but our target, as we'll see in future slides, these neurons can connect to parts of our brain. They can connect to different sensory receptors. They can connect to other things. But for the most part, we'll think about synapses or neurons connecting to other neurons. And we can actually see, it's the wrong way, we can actually see a visual of this over here. So here we have our initial neuron. So if this is what we've been talking about, this is the neuron that has our neuron or a neural signal. That's the action potential that travels along it. Between this neuron and the next neuron are our synapses. So we have the axon of this neuron, and then the synapse is the gap between them, and then the connection is to the dendrites. And again, a functional connection, but without actually touching. So we have the axon of the neuron that comes before the synapse. You'll see it also called a presynaptic neuron because it's coming ahead of the synapse. Then we have our synapse, this synaptic cleft, this gap between our two cells. And then afterwards, we have the dendrites of the synapse or of the neuron that comes after the synapse, or the postsynaptic neuron. So if we're talking about the presynaptic neuron, that's the one that comes before the synaptic cleft. And if we talk about the postsynaptic neuron, we're talking about the neuron that comes after that synaptic cleft. And so that's how we can refer to these two different neurons and how they connect together. And we've talked about the fact that there are millions of neurons in our bodies and they form lots of different connections. So we're simplifying this to look at just one neuron that connects to another neuron by way of this synapse. And so if we've talked about the fact that these aren't physical connection points, then what else is going on? And this is where, when we had talked about the two things that a neuron does, the first being that electrical signal that we just finished talking about, the second thing a neuron is responsible for is releasing chemical signals. And those chemical signals are how a neuron can communicate across this synaptic cleft, across this gap, to send a signal to the postsynaptic neuron. So they're sending this signal by way of chemical messengers. And these chemicals are called neurotransmitters. And we can talk about the specifics of how these neurotransmitters work. So our official definition is that a neurotransmitter is a chemical substance that carries a message across a synapse. And these neurotransmitters will either excite other neurons, and by excite, I mean trigger that next neuron to fire, so it can cause a new action potential in the next neuron, or it can inhibit their firing. So you could prevent the next neuron from firing. And we'll see some examples of this in just a few slides. So if we want to break this down into sort of the 5 basic steps of making use of a neurotransmitter, or they've called it the 5 stages of chemical communication, these are pretty logical, but we'll walk through each of them and then show a visual that goes with them. So in order to make use of neurotransmitters, you have to actually make neurotransmitters. So our presynaptic neuron, the neuron in front of the gap, has to make those neurotransmitters. So it's going to synthesize those neurotransmitters. And once it's made them, you have to put them somewhere. So stage 2 is storage. You're going to hold those neurotransmitters in a synaptic vessel. So you're going to put them into a container of some kind. I've also seen this called synaptic vesicles. Both are meaning the same basic thing, just a cell-type container holding onto our neurotransmitters. And so once you've made them and put them into storage, they're ready for use whenever that neuron needs to pass a signal on to the next neuron. So if an action potential has traveled the length of this neuron, and we want to pass that signal, that action potential, onto the next neuron, then we're going to release those neurotransmitters into the synaptic cleft. So stage 3 is actually releasing those neurotransmitters into the space between those 2 neurons. So into our synaptic cleft, or the gap between our 2 neurons. And so once our presynaptic neuron has dumped all of these neurotransmitters into the space between the cells, those neurotransmitters are going to start looking for somewhere to bind. And that just means that they're going to find a place that they fit so that they can attach to the next cell. And so this binding is going to involve receptor sites. And we have a visual on the next slide where we can see that certain neurotransmitters will only bind to very specific sites. The same way as when we were looking at action potentials and we had those ion channels that only worked with one particular ion, these receptor sites will only work with one type of neurotransmitter. So they can only stick to the place that they're supposed to stick. And once those neurotransmitters have bound to our postsynaptic neuron, once they've attached to that receptor site, they're going to cause some sort of change in that postsynaptic neuron. They're either going to excite it and cause an action potential, or they might inhibit it and reduce the chances of having an action potential happen. And once they've done that, they've kind of finished their job. And so the last step is to deactivate or stop that neurotransmitter signal. And so for this stage, you remove those neurotransmitters from the receptor sites. So now we can actually see the visuals going on with this. So we have our presynaptic neuron. So we can see that the signal travels down towards the end of the axon, those axon terminals, and this red circle is telling us that this is what we're looking at. So this is a blown up look at just one axon terminal connecting to the dendrite of our postsynaptic neuron. And so we can look at this blown up image here. And so we have that nerve impulse coming down the axon of our presynaptic neuron. And this impulse, that action potential, is going to tell this neuron that it's transmitting a signal across this gap. So our synaptic vesicles or vessels, these are holding our neurotransmitter. And when that signal comes over, it's going to cause those to bind to the end of our axon, to the axon terminal. And when it binds, it's going to push all of the neurotransmitters into the outside of the cell, so into this synaptic space. This is that extracellular space, this space outside of our cells. And so these little red dots represent our neurotransmitters. And these are going to spread out in this space between the two cells, and they should start binding to the postsynaptic membrane, so the cellular membrane of the next neuron. And specifically, they're going to be looking for the receptor that will fit with the neurotransmitter. And so up here, they have a very primitive cartoon showing a representation of what's going on. So here, this green type neurotransmitter can fit with this receptor because they are the same shape. Whereas if you have a different neurotransmitter, it has a different shape to it and it can't fit in that receptor. So these receptors only fit with very specific neurotransmitters. In reality, you're not going to see green spiky ones and purple square ones. What you would be seeing is that the molecules themselves have different shapes that stick to different neurotransmitter or to different membrane receptors in different ways. But this is a pretty good way of visualizing that at this point. And these are all of the steps. So synthesizing, storing, releasing, binding, and then deactivating at the end. And for deactivation, there's a couple of ways that we can do that. For deactivation, you can either remove these neurotransmitters from the receptor and then store them for later use so they could actually be cleaned up by different molecules that will stick them back into storage vesicles for use later on. Or you can break them down into their constituent parts and reuse them for other functions. So deactivation means that you either break them down or you reuptake them to reuse. Now, we have talked about inhibitory and excitatory action by neurotransmitters, but we're going to spend some time and go into a little bit more detail about that. So our inhibitory neurotransmitters, these are the ones that are preventing or making it less likely for our postsynaptic neuron to fire. So we're making it less likely for the next neuron to have an action potential. And so if we think back to our graph of what an action potential looks like, sort of like this, where we have our minus 70 and then positive 40 at the top. If we wanted to prevent an action potential, we actually have a time when action potentials are prevented. And that is during this refractory period when the cell at that point is overly negative. We talked about it being hyperpolarized, where it's more negative than our resting potential. And so our inhibitory transmitters actually do something very similar. So they will increase the resting potential of this postsynaptic neuron. So instead of having the normal resting potential, an inhibitory transmitter will drop the potential of that cell. So maybe instead of minus 70, maybe it's minus 80 now. And if your cell is sitting at minus 80, then it's going to take a lot more positive to trigger this action potential happening because we're starting so much more negative. So by being hyperpolarized or extra negative, these inhibitory neurotransmitters make it much harder for an action potential to occur. On the other hand, we can have an excitatory neurotransmitter. And so these are going to do the opposite. So instead of causing a more negative resting potential, an excitatory transmitter could cause the charge, the resting potential to rise. So it could become more positive, so closer to zero. So it would become less negative. So maybe it's going to be minus 60 now. Or if you get enough of this extra positive charge, which makes it less and less negative, this excitatory neurotransmitter may even trigger an action potential on its own. So it could cause enough of a rise in this charge to trigger this whole reaction and cause an action potential. So the excitatory neurotransmitters will make the charge closer to zero. So we'll bring it nearer to zero. The absolute magnitude gets smaller as we get closer to zero. And if it gets high enough, then we could trigger an action potential. Our inhibitory ones are making our are making our resting potential, make sure I say it right, more negative and making it less likely for the cell to fire. So we can look at it this way. Excitatory neurotransmitters, they are going to depolarize. So we're getting closer to zero. So the charge becomes less as we get closer to the absolute value of zero. We're going to depolarize that neuron's membrane. And when it happens like that, we can increase the likelihood of an action potential. So if you depolarize it enough, you can actually trigger an action potential. The inhibitory neurotransmitters, those are going to hyperpolarize that neuron's membrane. They are going to make it extra negative. The charge is going to get larger. And by doing that, they decrease the likelihood of an action potential because the resting potential becomes way more negative and it's harder for an action potential to occur. And so you can probably tell that if we're talking about these excitatory and inhibitory types of neurotransmitters, there are different types of neurotransmitters. And there are actually lots of different types of neurotransmitters. And we're only going to talk about a very specific subset of them, and specifically ones that tend to be very functional within the brain, just because those are the ones that we are going to encounter most. But just be aware that there are tons of them. And it's really neat because we see a lot of specialized neurotransmitters. And this is an intentional thing, or at least it seems to be. So if we're dumping these neurotransmitters into the spaces between these cells, if you think about it, you're actually putting these neurotransmitters outside of the cell with the intention of it being close enough to the next cell that the signal will travel. But there's nothing forcing those neurotransmitters to stay only between your presynaptic neuron and your postsynaptic neuron. There's nothing forcing it to stay in that small synaptic cleft. These neurotransmitters can spread out. They can travel. And so even though the concentration of them will stay highest where they're released, they're still going to move around a little bit. And in the brain, where you have tons of neurotransmitters and they're all very close together, you can run into issues where one neuron signaling the next neuron could actually trigger other neurons nearby. And so we have systems that try and help minimize some of this. And the way that this works is that different brain systems or different parts of our brain are sensitive to different neurotransmitters. So some parts of the brain will pay attention to one type of neurotransmitter, but they'll ignore other types. And this sensitivity and specialization helps avoid crosstalk between different areas of the brain. So it means that your parietal lobe can't accidentally affect your occipital lobe because there's specialized neurotransmitters and one part won't activate the other. So this is chemical specificity that helps make sure that there isn't noise transmitted between different parts of the brain that shouldn't be passed on. the next couple of slides are going to talk about a couple of very specific neurotransmitters. And so we're going to pick these ones because these are the ones that we end up talking about most, and they are most relevant in the parts of the brain that we're going to be focusing. So we're going to start with glutamate, which is sometimes also called glutamic acid. And I've included the chemical signals just as a reference. I'm not going to test you on the chemical signals. I just put them there because I find it interesting to see what they look like. But glutamate is an excitatory neurotransmitter. So when it's released, it's increasing the likelihood that the next neuron fires. And this is one that isn't specific to one particular part of the brain. This one is expressed throughout all of our brain. And it seems to be involved in the control of behaviors. So we'll see glutamate involved when we're going through processes involving specifically learning and memory, but there are lots of other roles as well. So glutamate is the one that we associate with controlling behavior, and specifically controlling our learning and memory type behaviors. For our next neurotransmitter, we have GABA, or gamma-aminobutyric acid. I will always include sort of both the long form and the short form, so you don't have to memorize the full proper name. But this one is an inhibitory neurotransmitter. So it's going to reduce the likelihood that the next neuron fires. And again, this one's expressed in the whole brain, so it's not specific to one particular area. And it's still involved in behavior, but this time around, instead of just being involved in learning and memory, it's more closely related to anxiety and motor control. So behaviors related to dealing with anxiety, as well as controlling your muscular movements. And GABA is one of the neurotransmitters whose imbalance is involved in Huntington's disease. So if you have Huntington's disease, it's a genetic disorder, and you would have incorrect amounts of this GABA that start influencing your ability to control your motor actions. So if it's not produced in the right quantities and in the right areas, it causes problems with coordination and movements and things like that, which is, it's a really unfortunate thing. I've talked about Huntington's disease in a class on genetics and behavior, and it's an interesting one. So if you wanted to take some time and look into it, definitely worth it for interest's sake. So our next neurotransmitter is acetylcholine. And this is another excitatory one, but this one isn't all parts of the brain. This one's more involved in functioning at synapses for muscle movement and memory. So it's going to really only function in areas when you're coordinating your muscle movement, but then also memory. So they don't have to necessarily be related for function. And acetylcholine seems to be what's driving memory loss in people who have Alzheimer's disease. So again, having incorrect quantities and locations of this particular neurotransmitter seems to influence your ability to form and retrieve memories, specifically for Alzheimer's disease. Our next specific neurotransmitter is norepinephrine. And now we're getting into some interesting things where norepinephrine is both excitatory and inhibitory depending on what part of the brain it's used in. So depending on the site, it could excite the next neuron or it might inhibit the next neuron. It all depends on which part of the brain we're looking at. And the functions change pretty dramatically, again, depending on what part of the brain we're looking at. So this one is involved in learning, memory, wakefulness, eating. Dysregulation of norepinephrine also seems to be involved in depression and panic disorders. So this one kind of covers the whole board. Just to give you an idea of some of the diversity and I guess the weirdness that happens within some of our neurotransmitters. Next we have serotonin, and this is one that you've probably heard about because serotonin is a mood regulator for the most part. Again, that's a little bit of an oversimplification. This one is mostly inhibitory, mostly because humans are humans, and sometimes the rules don't always apply. So there are certain areas where it is not inhibitory, but we'll stick with mostly inhibitory. It's going to function at different parts of the brain, and this is involved in mood, which is what it's well known for, but also in regulating your eating habits, sleep, and physiological arousal. And dysregulation of serotonin can be involved in depression or even eating disorders. And here, the things that it can be involved in kind of make sense, which is nice. So mood problems leading to depression, dysregulation of eating leading to eating disorders. And then our last one is another hopefully familiar face. This is dopamine. And dopamine is, again, excitatory and inhibitory depending on which part of the brain we're looking at. They can't all be nice and straightforward. But dopamine seems to be involved in voluntary movement. So when you decide to reach for something, or if you decide to move in a particular way, dopamine is usually involved. It's also involved in learning and motivation and feelings of pleasure. So a lot of what we know about dopamine is looking at the pleasure center of our brain and how we respond to positive things in our lives. And this one is tied into a lot of different problems. So dysregulation of dopamine can cause depression or Parkinson's disease or schizophrenia. So this one has lots of different functions and lots of ways that its improper presence can cause problems. But very interesting as well.